would like to introduce you to the FooD-STA project. It's the European Food Studies Training Alliance, a project that is funded in the Erasmus Plus Knowledge Alliance and will be active from January 2015 to December 2017. The coordinator of this project is Professor Gerhard Schleining from the BOKU in Vienna. Involved in this project are 7 universities, 3 food companies, and 11 multipliers and training providers. LVA is one of the training providers. The consortium is composed of universities and food companies. The universities are from France, Portugal, Germany, the UK and Italy, and Austria. The food companies from Portugal and Spain. And as associated member, Nestlé from Switzerland is also involved in the project activities of FooD-STA. Then there are the 11 multipliers and training providers I was talking before about. These are mainly industry associations, all related to the food sector, and training providers like LVA, or related to students and university networks like IFA. What is the vision of FooD-STA? Universities do a lot of knowledge transfer to students, and the knowledge should be useful for the food industry. But practice showed that there is a gap, that university knowledge and practical knowledge is not overlapping enough. The vision of FooD-STA is to close this gap by novel food study curricula that pay attention to food industry needs and are academic according to university needs. The FooD-STA centers that are founded in the running of these EU projects are physical hubs. These are institutes that are located in Vienna, in Hohenheim, in Paris, in Leeds, in Porto, in Teramo, Barcelona, and also in Athens. We would be very interested in your suggestions for topics that could be of interest for FooD-STA. We are designing curricula including the needs of industry, what topics are actually needed. So please contact us on office@iseki-food.net. Yes, today's lecture will be held by Professor Valerie Camel. She is a professor for analytical chemistry and food safety and the Department for Food Science and Technology at the AgroParisTech, is in a French graduate institute for science and engineering and associated to the Paris-Saclay University. Professor Camel was teaching there before and is now, as I said, at AgroParisTech. She is developing research projects concerning food contaminants. It's about all chemical contaminants you can imagine in foodstuffs and their metabolism and how they develop under the circumstances of treatment. She's an expert in chemical contaminants, mycotoxins, pesticides, migrants from packaging, and is also an expert as a member of the expert committee for the French National Agency for Food Safety. She is co-directing 3 master's degree programs on food safety, risk analysis, toxicology, and the assessment of management of toxicological issues for ecosystems and human health. I am pleased now to hand over the screen to Professor Camel. If you are ready, you may now take over.
Okay.
And I'm looking forward to your presentation.
Okay, thanks, Christine. So I will beginning the presentation. So hello, everybody. Thanks for participating to see this webinar. So I will present you the analysis of chemical trace contaminants in foods and discuss the performance and limits of the analytical methods. First of all, the content of the course of today. I will present the course in 4 parts. The first one, an introduction of the topics. Then I will present you the analytical approach that is commonly used and discuss the performance and limits of this approach, and then present future challenge for present time and future time. So chemical hazards in foods are always doing the covers of magazines. These are examples of recent front covers of French magazines, where you can see, even if it's written in French, I apologize for that, but you can see that there are always the topics on the front cover of the magazines about contaminants or pesticides in different types of foods. So it's a very big issue for consumers and mass media, and also, of course, for operators and academics that work in this topic. We can distinguish different classes of contaminants, and normally we speak of either contaminants or residues. So residues are supposed to be from substances that are authorized for specific treatment in the food chain, and there's specific regulation that authorize such treatments. So these are pesticides and veterinary residues. Then there's also contaminants from food materials. The most famous is bisphenol A. You also can find contaminants from processing. For example, acrylamide is very famous recently, or PAH or furan also. Then, of course, there's contaminant coming from the environment, for example, dioxins, PCB, or heavy metals. And then we also have natural substances, mainly mycotoxins, that are frequently encountered in foodstuffs. The regulation in Europe is evolving on this topic, and all the different classes are regulated, but more or less depending on the substances. So there's specific regulation related to pesticides and veterinary residues. Maybe I could reduce this. Yeah, okay. So these substances and this regulated, fixed maximal residual limits for these compounds. And then there's also a regulation for contaminants, processing contaminants, environmental contaminants, and also mycotoxins. So there's a generic regulation for all these compounds, and then there's some specific regulation added, or even recommendation in the case of acrylamides. So again, there's some maximum levels that are fixed and guide value in the case of acrylamide. And for food contact materials, there's also a specific regulation, and for all materials, and then specific regulation dedicated to the type of materials, especially for plastics, you can see there, which fix specific migration limits for the compounds we may find in the final food. So all the chemical hazards are not regulated at present time, but many substances are regulated. You may know that the operators in the food chain are, whatever the stage, are responsible in Europe for the food safety, especially the chemical food safety. And we have the chemical hazard that can enter the food chain at the different stages, from primary production to distribution, and even cooking and storage before consumption. And in Europe, we have an integrative approach for food safety, meaning that we speak from farm to fork or from stable to table. And this point is very important because there's monitoring of the quality of the foodstuff through the food chain, and this means that different types of samples are coming in the laboratory and need to be analyzed for the chemical hazards. So sampling and analysis is a very big issue, and these are, of course, key issues. So the samples coming from the different steps of the food chain are frequently taken for analysis. In the case of monitoring plants that are done by all the state members in Europe, and also when some notification of problem is given, there may be some control plans also to check the conformity of the foodstuffs. So these samples are dedicated to the foodstuff produced in Europe, but also to the foodstuff that are imported or the ones that are exported. And there's specific European recommendation for the sampling in the case of several contaminants, especially mycotoxin, for example, of PCB. These samples are coming to the lab, and you may have a network of labs with different type of lab. We can distinguish what we call the routine labs. These are the labs that will make most of the analysis of the samples in the case of monitoring plants or even control plants. And then there's some reference lab at the national level in the different state members that are dedicated to specific contaminant and specific foodstuffs. And these are have the missions of testing the proficiency of the routine labs and ensuring that they are up to date for performing the analysis. And they also made some development of methods. And then at the European scale, we have some reference labs also that will of course, develop methods, new methods, validate these methods, and also they will ensure that the reference lab are also proficient and have doing up-to-date methods. So these labs use validated methods. We will see that later, and that provide analytical results. And these results, of course, need to be reliable because they will be used either for risk assessment, especially by the European Authority about Food Safety, the EFSA, and also they will be used to check the conformity of the foodstuff and their compliance with the regulation when chemicals hazards are regulated, as I mentioned before. Especially then when there's some non-conformity, this will give rise to a notification on the RAS. The RAS being what we call the Rapid Alert System for Food and Feed, and it's a network that is working in Europe. And there's all information about the quality and the food safety is put on the rafts, and it's piloted by the European Commission. And you can see, for example, that last year, regarding foodstuff and feedstuff also, with contaminants and chemical hazard and the different type of contaminants, we had some notification about food contact materials related to heavy metals and migration compounds, with some serious notification due to the type of information. And then we also found, related to food, some mycotoxin, environmental contaminants, and also residues. And in the case of feed, of course, there's also monitoring, and some notification came related to mycotoxin again and environmental contaminants. So food and also feed matrices are highly complex, and we need to be aware of the structure, but also, of course, of the composition, looking at the chemical composition, especially they present macromolecular compounds such as proteins, sugars, fat matter, Also, you may find minerals, water, and so it's very important to have an idea of the type of food we're looking for the chemical hazard, especially the water content or the lipid content and other possible components that may be present and that may hinder the analysis of the chemical contaminants. Also, we need to have an idea of the interaction of the food and the contaminant, because we may have some contaminants that are easily accessible for analysis and others that are strongly bound to the food structure. This is, for example, the case of some mycotoxins that are considered as masked mycotoxin, because it's very difficult to have their analysis in some food, for example, cereals. And also we need to manage the heterogeneity and the high variability of the food and the great variety of food matrices we may encounter. In addition, the contaminants are present at very low levels. So there you have a graph showing the different content of components you may find in foodstuff. And regarding chemical hazards, we are at what we call trace and ultra-trace compounds, meaning that we are in the order of what we call 1 ppm, which is 1 part per million, and also 1 ppb, which is 1 part per billion. And the limits of most analytical methods are around 1 ppt, which is 1 part per trillion. So you can see that these are very low levels of the molecules we need to check. When we look at the analytical approach from sampling to the sample that is coming in the lab, this sample needs to be homogenized, and then there's one subsample that is taken, and this subsample is dedicated for analysis. And this subsample will be treated by different steps: sample treatment, then analysis, and then there's a great part of data treatment also that is performed in the lab to get the final results. And of course, due to the objective of the results that are either regulation, compliance of the foodstuff, or risk assessment, the reliability of the results need to be checked, and using validated methods, and also performing by laboratory that have shown their proficiency in using the method. So looking more deeply in the analytical process, there are different steps, such as what we call the treatment, and then the extraction, which is a core step of the treatment. These are performed by different techniques depending on the physical structure of the food. So some are dedicated for liquid foods and others for solid foods. And then after the extraction, we have the extraction using liquid phases. So there's always liquid extract, and these extracts most of the time are cleaned up, sometimes concentrated before being analyzed, most of the time with the target approach. We will see that later. And then there's the final data treatment. So these are all the analytical approach. In the past 20 or 30 years, different techniques have made relative to food analysis, especially relative to food contaminants. Among these techniques, I will focus on the coupling of liquid chromatography with mass spectrometry, which is now very crucial for the analysis of several and numerous contaminants. Also, the appearance, this coupling made at the beginning of the 19th. And then in the past 10 years, there was the appearance of what we call ultra-performance liquid chromatography, enabling liquid chromatography with high resolution and with very fast analysis time. And then also the appearance of high-resolution mass spectrometry. So this improvement in techniques enable now very fast acquisition analysis, improved capability of analysis for mature residue contaminants, and also high sensitivity of the analysis. So to illustrate the analytical approach, I took that from one publication. So we have to homogenize the food samples due to their large heterogeneity, and then extraction, clean up, sometime concentration, and then final analysis that is performed mainly using liquid chromatography coupled to mass spectrometry, or for some contaminants such as dioxins, for example, gas chromatography coupled to mass spectrometry using high resolution. In practice, the analytical methods are target-oriented, meaning that we know what we are looking for, and we will develop the methods to face the identification and quantification of these target contaminants. So looking at the contaminants, we will know the structure, the chemical structure of the contaminants we are looking for, and their property, especially the main property that are very important for the analytical method are the water solubility and also the octanol water distribution coefficient, and sometimes also the volatility and the acidity of the molecules. So these methods are referred to as methods that will look at what we call a priori non-contaminants, meaning that we will not see contaminants that are not targeted by the methods. For the development of methods, the food categories need also to be targeted to have an idea of the components that may be present and that may hinder the analysis of the target contaminants. So these are examples of food commodities categories given by the European Commission this year. You see that you have different categories and some representative foodstuff given in the table, and we will distinguish the commodity with a high water content, or that are quite acid, or with sugar, or with high content of lipids, and some animal origin also commodities. So now we will see the performance and limits that are achievable using the validation of these target-oriented methods. So typically we will get one chromatogram, and the first information that is very important is the retention time of the target contaminants. So we have an expected time of retention in the chromatographic column, and if we have a peak, we may suspect the presence of the contaminant. If there's no chromatographic peak, the contaminant is either not present or not detected by the method. When the one peak is present, it is very, very important to be sure that this is really the contaminant, because we may have some other components present in the food that coagulate and also give some chromatographic peak. So the first step for the validation is to confirm the presence of the contaminant that is as been target in real samples. So, and especially to be sure that there's no matrix effect to avoid, for example, false positive results. So as an example, we'll see on this slide the different steps for the compound confirmation. The acquisition mode, most of the time using mass spectrometry, can be done using different type of acquisition mode, but most of the time, because we are focusing on target contaminant, we will fix retention time windows with selected ions that are typical of the target contaminants in order to have selectivity and sensitivity of the method. And of course, we need to check for the stability of the retention time, and otherwise we cannot correctly detect the target contaminants. Then the peak is detected, and finally the quantification will be done by calibration using the target contaminant standard we have analyzed also similarly as a sample, and the identification is based on different criteria that are given in the regulation and that may differ a little bit depending on the type of regulation and the contaminants, but mainly these are the retention time, also the spectra and the fragment, the patterns obtained for the isotope, and also the relative abundance of the ions. So this is an example of confirmation of one compound in honey. You have there the extracted chromatogram for the main fragment that is of interest for this compound under the condition of analysis, of course. And there's some comparison with the same compound analyzed in standard. We can see that we have the same retention time and the same fragment is very important. And then we can have also the MSMS spectra of the standard and in the sample, showing that we have the same fragment and the relative abundance is also the same. And it's not visible there, but we can also look at the isotopic pattern, especially there because there's some chlorine atoms in the molecule. This gives information also about the chemical structure. So in this case, we have some good precision on the mass because we use high-resolution mass spectrometry, which is more informative than low-resolution mass spectrometry. An example to show the capability of high-resolution mass spectrometry. This is the case of one pesticide in black paper and the final analysis using UHPLC LC-HRMS. In this case, we have analysis with low resolution mass spectrometry. We look at specific transition that based on this fragment that is isolated, that is typical of the target compound. And then we look at two transitions that are also very representative of this target compound. We can see that there's one interferon coming from the matrix there, so the quantification is more problematic. And when we use high-resolution mass spectrometry, so another analysis with another instrument, we have a good precision on the mass we're looking for, and we can see that in that case we can isolate the mass and the peak of the compound, the target compounds, from the interferon of the matrix. And we have only the peak of the compounds, and we can confirm and quantify correctly the compounds in that case. So we have really increased selectivity and also sensitivity with high-resolution mass spectrometry. During the validation, we also need to have reliable quantification. Especially, we need to assess some performance of the method, especially the limit of quantification, and check its compliance with the maximum residual level of the target contaminants in the foodstuff. And we also need to assess what we call the trueness and the precision of the analysis. And we need to avoid matrix effect or to have some strategy when we observe matrix effects. So these are illustration of what we call the limit of quantification of analytical method. This is the level of the target contaminant in the sample that will give finally signal. So the signal is the intensity of the chromatographic peak for the retention time of the compound, a ratio of the signal to noise about 10, meaning that we are sure that at this level the target contaminant is present and that we can give reliable concentration for its level in the sample. So LOQ is very, very important as a performance of analytical method and must be assessed in the sample, not in solvents. There you have the trueness and precision that are illustrated. So the red points are individual analysis, single result of analysis of subsamples. And then when we get different subsamples of the same sample that are analysis, you can have a mean value for the level of the target contaminant in the sample. The dispersion of the different single results from the mean is the precision of the method. Of course, we need to reduce as much as possible the precision, but when we go to very, very low levels, the precision is not so small because it's difficult analysis. And then the difference between the mean value and the reference value is called a bias, meaning that if there's a bias, the method is not true because the mean value is different from the expected value of the sample. But if we are capable of identifying what is responsible for the bias, for example, it can be what we call the recovery, meaning that the extraction was not quantitative and we lose maybe 10% of the molecule during these steps because it was too complicated. Then if we have assessed the recovery, we can maintain this unquantitative extraction, but we can then, based on the value of the analysis, we can recover the real value. So if the bias is estimated and if it cannot be avoided, we can keep going and getting reliable results. To illustrate the limit of quantification of the method, and took the example of acrylamide, which is quite difficult for analysis in foodstuff, and it's quite not simple because it's a very, very small molecule, and so there are a lot of noise at this mass detected in mass spectrometry. So these are compilation of results of European labs that are dedicated for this analysis for different type of food commodities. You can see that there's a large discrepancy of the value for the limit of quantification of this molecule, ranging from around 5 or 10 micrograms per kilogram up to 100 micrograms per kilogram, and also the precision given by the interval there on the analysis are also very diverse depending on the food commodity. So you can see that the food type is very important for the assessment of the performance of the method, especially the limit of quantification. And also, of course, the lab method, especially the steps and also the proficiency of the lab, but also the instrument, because if you use high-resolution instruments, you will increase the capability of your analysis. Relative to quantification, we frequently have to deal with matrix effects, especially using liquid chromatography coupled to mass spectrometry, because during the ionization step, there's frequently what we call ion suppression or signal enhancement. So there you have the example on calibration curve of one pesticides. You have there the calibration curve, so it's external calibration curve using standards. You have the calibration curve in solvent that is frequently done to quantify the molecule in the final extract. And if we add the standard also in non-contaminated extract and get the slope, we can see that in orange extract and also tomato extract, we have deviation of the slope of the calibration curve that is over 10%, meaning that we have significant matrix effect in these two extracts, either with ion suppression there or with signal enhancement there, because the slope is either below or above the slope of the solvent curve. So these need to be assessed in order to have reliable quantification. When there are matrix effects, there are different strategies to overcome these effects in quantification. The first one that is very, very simple is to dilute the extract with the solvent, to dilute the interference from the matrix, and so to limit the effects on the ionization state. Of course, in the case of contaminants, this is not very useful because this will diminish the performance in terms of limit of quantification because you dilute the extract. Other strategies that can be used but are more complicated and more tedious are either to improve the cleanup step or to improve the chromatographic separation. For example, changing the stationary phase of the column or changing the composition of the mobile phase. Other strategies that are more simple to use and that is very frequently used for chemical hazards in complex matrices like food is to have internal standard with the stable isotope labeled molecule. So meaning that's the contaminant you are looking for, but with a stable isotope in the chemical structure, so that this labeled molecule is not present naturally in the food. So you will add it at the beginning, and you will see if there's some loss of signal, for example, some ion suppression. So it is supposed to mimic the behavior of the real contaminant that is non-labeled. So if there's some matrix effect on your target contaminant, you will see the same matrix effect on this labeled molecule, and so you can overcome the matrix effects. Other strategy, I will present you some example, is again to use high-resolution mass spectrometry analysis to increase selectivity and overcome matrix effects. And the other one is to perform what we call external matrix match calibration, meaning that instead of performing, I'll just go back to this slide, instead of performing external calibration solvent, we perform external calibration in the extract of the matrix to take into account the other compounds and these effects. So to illustrate the high-resolution mass spectrometry, you have there illustration of full-scan analysis of pesticides and then different compounds, and then you can isolate the exact mass of your compound of interest, and you see the chromatogram of considering only this mass, and there's only the compound of interest. The matrix match calibration, just rapidly, again, on microtoxin that give two fragments during ionization. And you can see that the mass spectra is quite different from the solvent extract and the matrix extract. We have the same fragment, but the relative intensity is very different. And so this can change the quantification and also the confirmation and the validity of the results. Finally, during validation, we need to check the applicability of the method also, especially the type of foods the method is dedicated to and the level range of the target contaminant. To finish, there are recent and future challenges related to this topic. The first one is to analyze simultaneously different contaminants that are target but that are coming from multi-class of contaminants. The other one is to analyze non-target contaminants and, of course, to identify new contaminants. So multi-class is a multi-residue and multi-class target analysis is quite recent. And just to give you some example taken from the literature, you have there a report analysis of more than 400 components from 3 classes of contaminants with the LQ below 10 microgram per kilogram using high-resolution mass spectrometry. And another one, again, with different classes of compounds with the limit of quantification below 10 microgram per kilogram. So this strategy, you use non-selective extraction, sometimes no cleanup, so very complex extract, and that require efficient separation, and that most of the time require high-resolution mass spectrometry. And the objective is to go to non-target approaches. So there are different strategies to have such approaches. One is to have the same approach as before, so extraction with quite no selectivity, some cleanup and analysis, and then to have the acquisition mode in the mass spectrometry that is independent of the data, meaning we acquire the data independent of the target of the molecule. For comparison, in the case of target analysis, we will select the ions and the fragments that are dependent on the target we are looking for. So we have more information. Of course, it's more complicated, but it's an approach to get non-target analysis. In fact, when we speak of non-target, we must distinguish 2 different strategies. The first one is not really non-target. It's what we call suspect screening, meaning that we have the different data acquired, and then we will suspect the presence of some known contaminants, and we will extract the exact mass of this contaminant, and we will see if there's a peak, and we will try to confirm the structure. So we use database that we have or that we will build to have this type of screening, enabling the identification of non-a priori target contaminants, but only of non-contaminants. The other one is also called retrospective screening, is to try to pick contaminant peak among the huge forest of peak we have, and avoiding irrelevant peaks, and to have based on the mass, to propose some molecular formula, and then to have additional information from fragmentation to try to propose a molecular formula for the new contaminant. So this is really a new identification of contaminants. So in conclusion, I will say that what we're resembling analytical chemists for contaminants in food in the past. In present, we are more better for the analysis of contaminants, but there's still a challenge to achieve, as I mentioned. And so in the future, we will be able to analyze more and more contaminants in multi-class and more rapidly. Thank you for your attention.
Thank you, Valerie, very much. Thank you. I got some questions during your presentation. I would like to forward them to you. Okay. They were posed from the audience. The first question is from Davide Rossi. Can you see it? It must be in the questions section of your panel.
Okay.
The question is, have you considered non-invasive surface energy analysis of external parts of foods as routes or for investigation of surface properties, changes after chemical treatment of this kind of food?
Well, it's very written. I cannot. I'm sorry, but I only have one line in the question. So,
okay, that's hard. But you can make the question part bigger by clicking a small icon on the right upper corner. A small icon with an arrow that makes this question field bigger. Can you find it?
yeah, I have the question. I'm sorry, but it's not the topic of my talk because I don't know what is meaning. But chemical treatment, I guess it can be with chlorine or maybe ozone or maybe other oxidants. I think I'm not sure. And the question is about the surface properties.
Yes.
But it's not.
That's how I understand it too.
I'm sorry.
Okay. So no problem. I have another questions for you. Simone Fritzen asks, can it happen that two or more components can be hidden under one peak, like overlapping?
Oh, sure. More components, meaning contaminants of target contaminants. Yeah, sure. So, but normally, if these are target contaminants, during the method development, you should have condition of separation in chromatography that ensure that the peaks are not overlapping. So normally it should be okay. Sometimes they may overlap because it's multi-residue, and then you can change and extract the fragments that are relevant for the molecules and isolate both compounds. So that's some strategy to overcome this.
Dr. Fritzen has another question. If a product is contaminated with mineral oil, is it possible to trace or identify which component of the product led to the contamination?
Well, mineral oil is not my topic neither, because it's quite particular. But normally, when you have the profile of the molecule that are present, you may be able to trace also the contamination initially. And that's I understood from the question. Is the question to trace the contamination in the food chain?
Yes, that's really a hard one. We have here a recent problem in Austria with mineral oils, and as far as I know, you can't distinguish really every single substance.
Yeah, normally, yeah.
Well, we have another question from Mr. Rossi. Which kind of changes in the structure occur in the time after treatment of food? Is it possible to detect these changes if occur despite the so low concentration of the contaminant? The excess of stability of food in the time respect the normal could be a parameter for a first assess of the contamination grade. This is a bit riddling for me. I forwarded you the question. Can you read it?
Yeah, I can. Well, again, it's change in the structure. So, meaning in the structure, it's not always contaminants. There can be a difference in degradation of proteins or lipids or other compounds that not give rise to contaminants. So, it's well, depending on the treatment, there are a lot of high temperature treatment that degrade the molecule in the food. Some of them are giving contaminants, but there's, again, we don't know a lot of things about that. So it's difficult to assess. Possible to detect the change.
A complex matter. Mm-hmm.
I'm not sure I understand. Sorry, the question, because it's
yes, it's a bit unclear.
Normally, when we look at what we call neoform contaminants that are coming from the processing, especially heated treatment, we have some information about the formation and the degradation of the compounds depending on the treatment. So normally there are sometimes some change in the food. In the case, for example, of acrylamide that I know quite well, we may have some coloration of the food. They give an indication of the degradation of some reactional scaling occurring in the food, and that can be an indicator of some acrylamide content of the food.
Mm-hmm. Well, we have another 4 questions. Mr. Ibrahim Abu Raida wants to know if you think that the detection of newer pesticides is a challenge.
New pesticides that are regulated, is that the question? I think so.
Mm-hmm.
Well, normally new pesticides that are on the market, the industry that synthesizes the pesticide should give the analytical method for these new pesticides. So we have normally some methods. This is a challenge in the sense of in routine lab, they do not have the proficiency of analyzing all new pesticides. Okay, so maybe we don't have the data for all these new pesticides, but in terms of analytical method, it's not for me a big challenge normally.
Mm-hmm. So there's another question from Brigitta Hengel. Mrs. Hengel wants to know, is it possible that LOQ value is different for the same food and same contaminants using ISO method for analysis?
The end of the question is using?
Is it possible that LOQ value is different for the same food and same contaminants using ISO method for analysis?
Oh, ISO
so standard methods, yes, okay. Standard.
Well, I think so, because it depends on the lab and also the instrument used by the lab. So if you speak, for example, like LC-HRMS analysis, depending on the type of analyzer you will have in MS, you won't have the same capability of the instrument. So it depends also on the lab and the system used. Yeah.
And Ms. Fernandez has another question. I'm just assigning it to you at this moment, so you could read it too. It's a long one. Is it possible to validate a method, for example, for pesticides without the MS detector? For instance, if we have an ECD detector, would we validate a method for pesticides or for PAHs with that detector? Or we need a MS to be sure that the compounds are well identified?
Well, for pesticides in routine analysis, normally MS detector is not always used, especially the ECD detector is very, very sensitive for some chlorinated, for example, or brominated molecules. But you need to check, at least for a few samples, that if you have a peak, that's really you need to confirm the structure of the molecule and the presence of the compounds. So it's not sufficient to have only one analysis with that type of detector. You need to confirm the presence, at least for the samples you are analyzing. Maybe not for all the analysis, but for the sample and the food of interest, you need to confirm the presence and the quantification. And in the case of pH, this detector is not appropriate. You should use other, because there's no chlorine, there's only carbon and hydrogen, so this is not appropriate for pH.
Okay, thank you. Another question from Dritan Topi. Hello, do you work with endocrine disruptors in food?
It's very vast, because endocrine disruptors, you have a different type of compounds. So some PCB are suspected to be endocrine disruptors, so I work on that. And also bisphenol A, for example, and other compounds coming from food contact material. So we do work on bisphenol A and other type of migrants coming from the packaging, actually, in our lab. Yeah. Other compounds. There are also some endocrine disruptors that are present naturally in the food, such as phytoestrogen, for example, that are not considered as contaminants, but they are biology active, in the same way. So it's a very vast domain.
Yes, it's a vast domain and not homogeneous. Yes. So these were the questions that arrived in my question box during your lecture. Thank you very much to everybody who attended and was so interested in this lecture and posed all these questions. I may also forward you the thank you from David Rossi, who had his question. Thank you also from Mrs. Dritan Topi. And I'm sure I speak on behalf of the whole audience when I say thank you to you too. Mr. Topi, thank you for this. Thank you very much, Valerie. It was a very interesting lecture. I'm sure that the audience could have a lot of information from it.
Thank you for all the participants, because it was great for me also to have so many questions and so many participants. And I look forward to having their feeling about the presentation also, because it's interesting for me also.
Yes, very nice of you to say this. It's always a bit hard to do the online webinars satisfying, I must say. So what I wanted to show you for finalization is the website of European FooD-STA. This is the website where we will publish the recording and also some material under this point events, webinars. So everybody who missed the webinar or wants to get to it back later can do so on our website. So thank you very much for your attendance. Thank you very much for being with us. Thank you, Valerie. Have a nice time. Enjoy the season. Goodbye.
Goodbye. Thanks all.